At once. Go. All right, good. We can see your screen. And we're just going to wait a couple seconds here until 9:15 on the dot. And Oscar, you are still unmuted. All right, Zach, go ahead whenever you are ready.
All right, thank you. Hello, everyone. My name is Zachary Griebenow, and today I will be talking to you about my endeavors in the realm of trying to infer a phylogeny for ants of the subfamily Leptanillinae. First off, I'd like to thank everyone who has helped me make this project possible, and particularly those who loaned or gave me specimens that were required for this research. I will start off by introducing Leptanillinae ants in a general sense, and then discuss how I went about inferring their phylogeny, mostly with a phylogenomic approach, but also using Bayesian total evidence inference to try to resolve the placement of terminals for which molecular data are unavailable. And finally, I'll give a quick preview of my embryonic efforts at divergence dating for the clade. So I like to introduce Leptanillinae ants by saying that they are to other ants as monotremes are to other mammals. Leptanillinae ants are sister to nearly all other ants and show a panoply of very derived characteristics juxtaposed with some that are uniquely ancestral among the ants. In terms of biogeography and biology, all Leptanillinae exist in the Old World tropics and subtropics. This will come up again later. And they are specialized predators of geophilomorph centipedes. The wingless female workers almost never come above the surface of the ground, and so most of the specimens we have are male, since the males have wings. This is the case to the point that many major subclades within the Leptanillinae are known only from male specimens, and this acquisition bias is the opposite of what we see in most ants. As you can see from this picture, male phenotype is quite diverse across the Leptanillinae, and this diversity is especially extreme with respect to the male genitalia, which show morphological innovations that are sometimes not only unique among the ants, but unique among the Hymenoptera as a whole. To understand this and other evolutionary phenomena within the Leptanillinae, we of course need a phylogeny. I mainly went about inferring a phylogeny for the Leptanillinae using ultra-conserved elements. With data at this scale, one of my main concerns was that my results would be affected by systematic bias due to model misfit. So, to work around that, I implemented phylogenomic inference under a number of different statistical approaches. Maximum likelihood, of course, with both by locus and within locus partitioning schemes, also Bayesian approach, and a two-step coalescent approach to try to accommodate the effects of gene tree discordance. I also repeated these analyses with a number of different UCE matrices curated to compensate for different potential sources of informational bias. And I trimmed each of these matrices for misalignment using spruce up under 4 different log normal cutoff thresholds. If you want more detail on this, I would suggest you ask about it later in the interest of time. So, was the phylogeny of the Leptanillinae robust to these analytical perturbations and different data curation regimes? To be brief, yes. There's not really a visually appealing way to show this, but the relationships among the major clades of the Leptanillinae, as seen here, were stable and strongly supported for the most part, regardless of what I threw at the phylogeny. The only notable exception being the relationships among the 4 subclades of the genus Protonilla, shown here in various shades of pink. So this is all quite reassuring for me, but it's also confirmatory and dull in my opinion. So with that robust phylogeny in hand, we can move on to more interesting questions. For example, there is the reality that molecular data are unavailable for several Leptanillinae lineages. And for the 4 of them, shown here, only male morphology is known, no molecular data, but that male morphology is so weird that they can't be intuitively, these terminals cannot be intuitively placed in the Leptanillinae phylogeny. This is especially the case for the species Leptanilla anomala, which was so bizarre that it was initially classified as Hymenoptera incertae before being correctly identified as a male ant, and in particular a leptanilline, before I finally placed the species in the genus Leptanilla, based on the results that I'm about to present to you. So to try to resolve the respective phylogenetic positions of these oddballs, I took a Bayesian total evidence approach. I used male morphological data discretized in a binary fashion and coded in a non-additive fashion, combined with a reduced alignment of 58 UCEs, subsampled so that this data set would be computationally tractable for a Bayesian approach. And then I also used a backbone topology consisting of 72 terminals for which both male morphology and UCEs are available, with this backbone topology conforming to the robust phylogeny that I showed you earlier. I implemented all of this in Rev Bayes and did not partition either the molecular or morphological data set due to misbehavior in the MCMC when I tried to do that. All 4 morphology-only terminals were included so that their morphological data could respectively inform their phylogenetic positions. So the results that I got with respect to these 2 terminals, the data that I used under the model that I used could not confidently resolve their phylogenetic positions within the Leptanillinae, which is disappointing, but not surprising. The morphological data are apparently just insufficient to do that in a probabilistic framework. For Leptanilla anomala, however, this terminal is confidently recovered within the Leptanilla havilandii species group, with Bayesian posterior probability being quite high. Although I will admit that its exact position within that clade remains much more uncertain. And finally, Leptanilla astylina is recovered as sister to the undescribed morphospecies Leptanilla TH07 with moderate support. And this placement doesn't make a whole lot of sense, either from a morphological or biogeographic standpoint. So I suspect that this may be an artifact resulting from so-called bare branch attraction. This is a phenomenon in Bayesian total evidence inference, wherein an aberrant terminal for which only male morphology is available, such as Leptanilla astylina, shown here on the left, is artifactually attracted to a terminal like Leptanilla TH07, for which both molecular and morphological data are available, but a large chunk of the morphological data are missing. And in the case of Leptanilla TH07, this is quite literally the case because the only available specimen is lacking the male genitalia. So I could not code morphological data for this anatomical region. So in the end, we are presented with a range of scenarios for Bayesian total evidence inference. We have cases where this approach cannot resolve the phylogenetic position of terminals for which molecular data are unavailable, a case in which it can be confidently resolved, and a case in which the recovered phylogenetic position is being misled by analytical artifacts. So, with all of that over with, I can talk about my early preliminary efforts at trying to infer a chronogram for the Leptanillinae. Dating the phylogeny of the Leptanillinae would be interesting for a variety of reasons, but to me, the most interesting one is trying to answer the question of why there are no Leptanillinae in the Americas. In the Neotropics, we have similar looking but very distantly related ants filling the same niche as seen here. The Leptanillinae could have crossed into the Americas in the late Cretaceous or early Paleogene using the land bridges shown here, but it's evident that they didn't. So was that because at the time the Leptanillinae did not exist yet, or some other reason? This could only really, we could only start to begin answering this question with a dated phylogeny. A big challenge here is that there are no Leptanillinae fossils, and so any dated phylogeny for the Leptanillinae, inferred under a clock model, of course, would have to have that clock model informed by observations of other fossil ants. Thankfully, the fossil record of the rest of the Formicidae is quite rich. So I plan to infer a chronogram for the Leptanillinae under an unresolved fossilized birth-death process. I will have a set of extant terminals, including 79 leptanilines, 72 non-leptaniline ants, and then 11 other outgroups. The most inclusive clade to which all of these terminals would belong is called the Ubiquiliata, and the clock model for this analysis will be informed by stratigraphic data for 162 fossil observations from across the Ubiquiliata, and with their respective phylogenetic positions constrained according to prior taxonomic knowledge. Because of how computationally intensive and complicated this will be, I haven't made it very far, but I do have a well-justified prior on my origin time, phi, which in this case necessarily corresponds to the crown age of the Ubiquiliata. And you can see this prior probability distribution here with millions of years ago on the x-axis. I inferred this without recourse to a phylogeny using a Bayesian-Brownian bridge from 1605 paleobiology database records of Ubiquiliata and an estimate of the extant diversity of this clade. But again, this is quite nascent, and I welcome any advice or criticism about the approach that I plan to take in inferring inferring a chronogram for the Leptanillinae. And with that, I will take your questions.
Excellent. We do have enough time for a couple of questions. We already have one in the chat, so let me read that out to you. And we have two in the chat. Since only males are being recovered and they are morphologically strange, is it possible that some of these specimens are not actually leptanillinae ants?
Let's see, let me stop sharing. Sorry. At this point, I would say no, because I have a very clear sense morphologically of what the diagnosis for the Leptanillinae is. Granted, there are cases in which, for instance, Leptanilla palauensis, one of those morphology-only terminals, that one was weakly recovered outside of the Leptanillinae, but the support is so weak that I really doubt that that is in fact the case. And then also how many morphological characters I used, that would be, I believe it was 64. Yes, 64 binary, non-additive.
So do you think that one taxon or that one tip was being dragged outside of the main group by its age information, or do you think it's just sort of artifactual?
Well, this was a total evidence analysis that did not involve any fossils, so all of these are extant. So I think that is stochastic error, honestly.
Okay. Sounds good. We've got to get our next speaker up. Thank you so much for this, Zachary. This is a really good talk. Linford, it looks like you are now here. Can I get you to try sharing your screen? Okay, here we go. Good, we are able to see that. Do you want to try unmuting?
Okay, hello. Can you hear
Good,
me?
we can hear you, yes.
Okay,
We'll just wait until right at 9:30 to get started in case anybody's straggling as they're coming in, and go ahead and start whenever you're ready.
okay. All right. You mean I can start anytime I'm ready?
Yep, go right ahead.
Oh, okay. Yeah, my name is Linford, Otoo, at Wake Forest University, second year PhD student, and my work is the genetic signatures of Emergent P8, VP4, Reassortant DS1, like human rotaviruses. Oh, okay. Okay. All right. Throughout the history of mankind, viruses have proven to be an important human pathogen because they are the major causes of various pandemics, such as the recent influenza pandemic in 1918, and also the most recent 2020 COVID-19 pandemic. Now, the virus I'm particularly interested in is rotaviruses, which have also shown to be important human pathogen because they are the major causes of because they cause severe watery diarrhea and vomiting in children, causes more than 200,000 deaths per year, and usually prominent in developing countries. And these viruses do not just affect children, but also affect a wide range of other organisms as well, such as horses, bats, dogs, pigs, cats, mice, cattle, etc. The good thing is that they are live attenuated vaccine called Rotarex and Rotatec, and it's available in more than 60 countries worldwide. However, these vaccines have shown to underperform in some developing countries because of malnutrition, etc., and also cannot be used in immunocompromised children because these children are not able to elicit immune response against the live attenuated vaccine. And the vaccines have also shown not to protect against some emergent strains. Therefore, it's important to characterize the genetic diversity and evolutionary dynamics of rotaviruses, which would help to improve the next generation vaccine design. Now, so this structure of the rotavirus particle is made up of is a triple layer particle. So the outside, okay, I hope you can see the mouse. Okay, so the outer capsid protein is made up of VP7, which is the yellow, and the red is VP4 with the outer capsid protein. Then the intermediate layer, which is the green, which is VP6. Then the innermost layer, VP2, and the VP2 houses the 11 segmented double-stranded RNA, which is not shown. So these are the 11 segments separated by gel electrophoresis, and these are their various functions. So the VP1 is the RNA-dependent polymerase, and the VP2 is the core shell protein, as I showed you earlier. Now, in order to in order to classify the rotavirus, Yella classified the new system by using the entire segment. So they actually assigned a letter to each of the various function or segment. So the R here signifies the RNA dependent polymer. The C is the core. The M is the methyl transferase of VP3. And a genotype number was assigned to them based on the percentage identity cutoff value. So 28 means that the genotype of R, there are 28 different types of RNA-dependent polymerase of rotavirus, or VP1. So these letters were brought together to form the genotype constellation in order to classify the virus properly. Now, paper by Martinez 2009 showed that most of the rotavirus that infected humans were either green or red, mostly. Like mostly it's either green or red. So the all red is the pure DS1 like, and the all green is WA-like genogroup. However, there was another type that was found, another combination, which is called the intergenal group reassortment, which is a combination of both red and green together within the constellation. And that is where my work is mainly focused on. Now, so before 2013, either the parent strain, either all blue or all red, were known to be more fit and were circulating in the population. As I said, either more green or more red. However, but we never really saw the reassortment happen before 2013, probably because it wasn't ever reported. I mean, some of them were reported, but they were very, very minimal. And it was hypothesized that they were less fit, and because of suboptimal protein-protein interaction between the red and the green, and also RNA-RNA, suboptimal RNA-RNA interactions as well. And also there were low co-infection rates that could lead to their rare detection. So before 2013, the reassortment was really, the reassortants were really rare. However, from 2013, it showed that they became more fit as more of them were being detected globally. So this is some few papers that showed the emergence of the IGRs, that is the rotavirus with mixed genomes from both strains, and it was even found to even infect vaccinated children. That makes it very important for us to study the IGRs in humans. So this where my research question comes in: how similar or different are the P8DS1 reassortment different from the typical P4DS1? And also, if they are different, are there unique changes that that distinguishes the reassortants from the wild type strains? And how are these unique changes going to help us to understand the emergence of these novel reassortants circulating in the globe? So in order to do that, I designed a phylogenetic analysis. First of all, I cumulated all the published strains way back from 2000 all the way to 2025, sorry. And then the inclusion criteria is that it's supposed to be published in humans, and the exclusion criteria is no animal and no other rotavirus strains, such as rotavirus B, C, D, no, only rotavirus A, because that affects like 99% of the human population. And also design a constellation table using what I showed you, that the letters and their respective genotypes in a color code, but I'm not going to show that. Then also perform multiple sequence alignment and perform phylogenetic analysis using neighbor joining method. Now, so this how the typical P8W1 strains, that is all green, and the typical P4DS1 strain, which is all red. However, with the reassortment, we can see both, right? So now this is where my question comes in. Is the red found in the reassortant the same as the red found in the typical P4DS1 strains? Is it the same or totally different? And also the P8 in the reassortant, the green P8 right here, is it the same? Is it genetically the same or different from the typical P8WA1 strains here? So that's the my work actually. So with the phylogeny of the VP7, this for G2, then G1 and G9, then these two are G3, and which showed differences, and these two here show the G8. So with the G3, I realized that the G3 that are involved in reassortment or that were reassortant were clustered totally different or separated from the non-IGL G3. That means that G3 involved that are not, or let me say, that are typical strains were totally different from the GL that caused this, that were involved in reassortment. The same for the G8. So G8 shows some divergence, showing that we have IGR specific G8 that is different from the G8 that are found in the typical G8 human strain. And I did the same thing for all the 11 segments. So with the VP4, which is the protease spike, which are involved in interacting with the cell type, with the surface of the cell for infection. So the light green here, so these are all the reassortant strains, which we can clearly see a total separation from the typical VP4 in the typical strains. We also did the same thing for VP6. So now, anytime you see the yellow right here, shows that there is separation of the reassortants from the typical strains. And we did the same thing for VP1, VP2, VP3. So as we can see, the yellow clade here clearly shows that there's a distinction between the reassortant from the typical strains. We did the same thing for the NSP. The NSP are non-structural proteins, which does not form part of the structure of the virus. And we see it all the way through, except for NSP5. NSP5 did not show any divergence. And this typically shows that yeah, so the NSP5 is the same. So the NSP5 is same for both the reassortant and the typical strains as well. So in conclusion, got to understand that the PHDS1 reassortant strains are genetically distinct from the typical P4DS1. So now that we know that they are significantly different, or sorry, they are genetically different, we try to determine the unique changes that differentiates the IGRs from the typical strains. So I perform multiple sequence alignment, then I use the camera software to determine where these changes are, and also correlate it with biological importance, and also find its specific location on the virion structure. Right, so with the VP1, we found the mutation on the N-terminal domain, which is really close to interacting with the next structure, which is the VP2, and also found two amino acid mutations in the polymerase domain that could account to why we see these viruses, this new strain infecting even children, because it probably have high polymerase activity because of this mutation. With the VP2, which is the inner, which is the inner core protein, we found only one amino acid change, and the most what's really interesting is found within the section where it interacts very closely to the VP1, because the VP2 has direct interaction with VP1. VP2 has direct interaction with VP1. VP1 is the polymerase. VP2 is the inner core protein. So you can see that it's very close to the so the section we see is the VP1, and there are various VP1 surrounding the VP2. So probably there's a higher interaction between, or a better interaction between VP1 and VP2 because of this change in amino acid there. And the VP3 found this various interaction for the methyl transferase, which could probably be more efficient. That is why we see this reassortant in circulation. The same for VP4. VP4 is the as I said earlier, is the protein that interacts directly with the cell for interaction, and we found 3 amino acid in the VP8. So the VP8 kind of interacts directly with the receptor, whilst the VP5 section is embedded within the virion structure and interacts with the VP7 and VP6 as well. And we found mutations on all 3 specific domains, clearly showing that probably because of these changes at the surface of the VP4, it could interact better with the cell surface. And also we found this K24060 directly interacting with the VP7, and also found these two amino acids, I58F and the K62R, interacting directly with the VP6. And it could possibly mean that these amino acid changes facilitated like proper interaction or high level interaction with the other proteins that make up the variant.
Linford, that is the end of your time.
Okay.
All right. Thank you for your talk. So this is the last talk in our session today. So we do have some time if you would like to hang out. We're going to go ahead and stop recording.